Okay, welcome everyone to this screencast of part 2 of our lecture on the respiratory system. You'll recall that we left off in part 1 talking about how oxygen is transported by the blood from the lungs to the peripheral tissues of the body. And we mentioned that there were two methods of transport. We said that there was a tiny amount or a small amount of oxygen that was transported in the plasma of the blood. However, we mentioned that oxygen does not mix very well with the plasma of the blood because it is a nonpolar molecule, and the plasma is made up primarily of water, which is a polar molecule. So polar molecules and nonpolar molecules don't mix very well. So not very much oxygen mixes with the plasma of the blood, and therefore not very much is transported in the liquid portion of the blood to plasma. So most of the oxygen moves from the plasma once it enters the blood and passes into the red blood cells, where it combines with hemoglobin, and the majority of that oxygen is then transported on the red blood cells via the hemoglobin. All right. So today what we want to do is turn our attention to the second respiratory gas, which is carbon dioxide. We want to talk about how carbon dioxide is transported via the blood, this time from the peripheral tissues back toward the lungs. And as we take a look at this, we're going to see that there are actually 4 methods by which the carbon dioxide is transported. We're going to take a look at each one of those different methods here. So let's begin. And our first method of transport that we take a look at is, again, a small amount of carbon dioxide can be dissolved in the plasma. So we're out in the peripheral tissues now, and the cells of the peripheral tissues are active cells, and they're carrying out cellular respiration. And we understand that a byproduct of cellular respiration is carbon dioxide. So the cells have to be able to get rid of this carbon dioxide in order to be able to continue their activity. So what happens? The carbon dioxide inside the cells builds up into higher concentrations and eventually follows its concentration gradient and diffuses out of the cells and into the interstitial fluid. As the interstitial fluid begins to accumulate carbon dioxide and concentrations build up once again, now the carbon dioxide again follows its concentration gradient and moves from the interstitial fluid across the wall of the capillary bed and into the blood plasma. Now, once again, just like with oxygen, carbon dioxide is a nonpolar molecule. So when it enters the blood plasma, it does not mix very well with the plasma. So a small amount of carbon dioxide will be dissolved in the plasma, but the majority of that carbon dioxide will continue to move from the plasma, and where does it go? Most of that carbon dioxide is going to enter the red blood cell. So it's going to continue to follow its concentration gradients because it doesn't mix very well with the water molecules in the plasma. Now, once the carbon dioxide enters the red blood cells, it is converted into bicarbonate ions. And we can see over here on the right-hand side, here's a bicarbonate ion. It consists of a hydrogen atom, a carbon atom, and 3 oxygen atoms. Now, what makes this an ion? Because traditionally, whenever we think of ions, we think of atoms that have either a positive or negative charge associated with them. Now, why do they have that positive or negative charge associated with them? It's because they either have more protons than electrons, in which case they'd have a positive charge, or they have more electrons than protons, in which case they would have a negative charge. But here, we're looking at a group of atoms, and if we take this group of atoms together, we can see this negative sign we have up here. That means this is a negatively charged group of atoms. That's because this group of atoms actually has associated with it more electrons than it does protons, so it takes on an overall net negative charge. So we could still consider this as being an ion, and we call it the bicarbonate ion. So most of the carbon dioxide enters the red blood cells and forms bicarbonate ions. The majority of those bicarbonate ions then, once they form, will leave the red blood cells and now move back out into the plasma, where they'll travel in the plasma as sodium bicarbonate ions. Now, sodium bicarbonate is an ionic compound, and I should have a little plus sign above the sodium here and a little negative sign above the bicarbonate ions. And that's because what's going to happen? Whenever this sodium bicarbonate mixes with water, it's going to separate into sodium ions and bicarbonate ions. Well, these are now positively and negatively charged, and so they can be picked up by the water molecules and transported in the blood plasma, mixed in with the water molecules and held separate from one another, dissolved in the plasma. So where carbon dioxide could not be dissolved in the plasma, these bicarbonate ions in the form of sodium bicarbonate can now be dissolved in the plasma and carried more easily by the water molecules in the plasma. A third method of transport of the carbon dioxide is some of the bicarbonate ions that were formed in the red blood cell will remain in the red blood cells, and they will attach to potassium ions, which we can also find in the red blood cell. Now remember, potassium ions are also positively charged, so these negatively charged bicarbonate ions will be attracted to the positively charged potassium ions to form potassium bicarbonate. So some of the bicarbonate ions in the red blood cells will be picked up by potassium to form potassium bicarbonate. And then our final method of transport is any carbon dioxide that hasn't been, once inside the red blood cell, that hasn't been converted into a bicarbonate ion will now be able to combine with hemoglobin. And the carbon dioxide will then be carried by hemoglobin as carbaminohemoglobin. And here's the formula for carbaminohemoglobin. Okay, let's try to break this out. On the next slide, you have an open slide. And so we're going to try to fill in that open slide and give you the opportunity to kind of break this out and diagram it so we can understand a little bit better. Because I think whenever we diagram things out, we can understand the written word a little bit better. All right, so let's take a look at this cell here now. Okay, and let me just make an adjustment here. Okay, let's take a look at this slide, I guess I should say. And on this slide, we're going to have, this is going to be a capillary bed. Okay, so with one big giant red blood cell in the center of the capillary bed. So in our capillary bed, this is going to be the arterial side, let's say, just for the sake of discussion, the arterial side here on the left, and over here on the right, we're going to call this the venous side of the arterial of the capillary bed. So blood would be flowing from the arterial side across the capillary bed to the venous side. Now down here, these are going to represent the cells of the peripheral tissues right here, the active cells. Remember, they're carrying out cellular respiration. Each one of these cells, through cellular respiration, is producing ATP and a waste product, carbon dioxide. And as we mentioned, as that carbon dioxide is produced, it accumulates in the cells, and it begins to move along its concentration gradient. So the carbon dioxide is going to move out of the cells and into the interstitial fluid. Now the carbon dioxide is in a higher concentration in the interstitial fluid than it is inside the capillary bed. So what's going to happen? The carbon dioxide is going to continue to move along its concentration gradient. It's going to pass through the wall of the capillary bed and enter the plasma. And we can see here the carbon dioxide now in the plasma. Now, a small amount of that carbon dioxide will be carried off along with the plasma. But as we said, carbon dioxide does not mix very well with water molecules, so not very much is going to remain in the plasma. And what's going to happen is the carbon dioxide now is going to continue to diffuse along its concentration gradient. So where does it go next? Well, the carbon dioxide is now going to move into the red blood cell along its concentration gradient from where it's in a higher concentration to where it is in a lower concentration inside the red blood cell. Once that carbon dioxide enters the red blood cell, it's now going to combine with water. And when it combines with water, it's going to form an acid called carbonic acid. Now, we'll just stop here for a second before we go any further, and we might ask ourselves the question, well, why does the carbon dioxide combine with water inside the red blood cell? Why doesn't it combine with water out here in the plasma? I mean, plasma is 90% water, right? Why doesn't it combine with water in the plasma? Why does it wait till it gets inside the red blood cell? The answer to that question is because inside the red blood cell there is an enzyme, and that enzyme is called carbonic anhydrase. Carbonic anhydrase catalyzes the interaction between the carbon dioxide and the water molecule to form carbonic acid. Okay, so that doesn't happen out in the plasma because there's no carbonic anhydrase. There's no enzyme out there. So now, once this carbonic acid forms, it is an acid, so what do acids do? Acids dissociate to release hydrogen ions into solution. So as almost immediately, as soon as this carbonic acid forms, it's going to dissociate to release hydrogen ions, as we can see here, into the cytoplasm of the red blood cell. Now, as the hydrogen ions are released, what's left over? Well, you can see we have H2, and if we release a hydrogen ion, there's one of our H's. That leaves another H left over, plus the CO3. And HCO3 forms our bicarbonate ion. So this is how the bicarbonate ions get formed inside the red blood cell once the carbon dioxide enters. Okay, let's focus on these bicarbonate ions for a minute. So what happens to the bicarbonate ions? Well, some of the bicarbonate ions, once they're formed, they are going to seek out potassium, and they're going to combine with potassium to form potassium bicarbonate. Remember, this is one of the ways that we said that the carbon dioxide is transported inside the red blood cell as potassium bicarbonate. But there's not enough potassium inside this red blood cell to link onto and collect up all of these bicarbonate ions to be able to transport them. So what happens? The remaining, and that's really the bulk of the bicarbonate ions, are now going to accumulate into higher concentrations inside the red blood cell. And what happens? Well, just like always, substances always want to move from where they're in high concentration to where they're in low concentration. This time, the bicarbonate ions are in a high concentration in the cytoplasm of the red blood cell and in a lower concentration out here in the plasma. So the bicarbonate ions will want to move from the red blood cell out into the plasma. To facilitate that movement, there is a transporter protein, and we'll just use that. We'll show brackets here to represent that transporter protein. That transporter protein is called the band 3 protein. The band 3 protein, when the bicarbonate ions form, will transport those bicarbonate ions, once again, along their concentration gradient from where they're in high amount inside the red blood cell to where they're in lower concentration outside the red blood cell. Once the bicarbonate ions enter the plasma, the bicarbonate ions will combine with sodium ions. Remember, sodium ions, think back to your discussion on neurons and how action potentials are formed. And we find that some cells don't like sodium ions, so they don't allow sodium ions whenever they're in a higher concentration to move along their concentration gradient from where they're in high amount to where they're in low amount. So sodium ions tend to accumulate along the plasma membrane on the outside of these kinds of cells. And red blood cells are no different than those neurons. They don't like the sodium ions to come in. They don't have a lot of channels for them to come in. So sodium ions build up in a high concentration outside the red blood cell. So we can see here that as the bicarbonate ions come out, they will link on to sodium ions to form sodium bicarbonate. And this represents another method of transport that we've talked about in the previous slide. So the bicarbonate ions will combine with sodium to form sodium bicarbonate, and these are opposite charged, so the polar water molecules will be able to pick this up and separate the sodium and the bicarbonate ions from one another and carry them along in the plasma. And again, they're carried along with the water molecules. Okay, now the only problem that we have here is that as these bicarbonate ions are leaving, remember the bicarbonate ions are negatively charged. And they were formed whenever hydrogen ions were liberated from the carbonic acid. So now what's going to happen is if we lose all of these bicarbonate ions from the red blood cell, we're going to start to have an imbalance of positive and negative charges. So what's going to happen is because the hydrogen ions are going to be retained here, and we'll talk more about what happens to them in just a bit, these hydrogen ions are going to be retained, but these bicarbonate ions, which are negatively charged, are going to be lost. So what's going to happen is very quickly, the red blood cell, if we don't do something, is going to begin to have an imbalance of positive and negative charges. And we don't want that to happen. So we got to balance out the positive and negative charges again. To help with that, also along the outside of the plasma membrane of the red blood cell, there are chloride ions from the salt, sodium chloride. These chloride ions are negatively charged. So what happens is the chloride ions now will be shifted once again by the band 3 protein from the blood plasma through the plasma membrane of the red blood cell and into the inside of the red blood cell. So for every bicarbonate ion that comes out, a chloride ion moves in. This helps to maintain the balance of positive and negative charges. Bicarbonate ions out, chloride ions in. This process of replacing the bicarbonate ions with the chloride ions is referred to as the chloride shift. So the chloride shift refers to the exchange that we've just talked about of the bicarbonate ions and the chloride ions across the membrane of the red blood cells. And again, what's the purpose of that? It is to maintain proper ion charge balances inside the red blood cell. Okay, next up, we've seen what happens to the bicarbonate ions. What about these hydrogen ions? What happens to the hydrogen ions? Because as the hydrogen ions are released, the pH of this red blood cell is going to drop. And these hydrogen ions can go around and they can cause damage to proteins. So we don't want the proteins to be damaged. So what's going to have to happen is those hydrogen ions are going to have to be taken up out of solution so they can't run around and cause damage to other proteins in the cell. So who picks up these hydrogen ions? Well, That's one of the jobs of hemoglobin. You know, traditionally we think of hemoglobin as being the oxygen, the red pigment that transports oxygen in the red blood cells. But hemoglobin also has the ability to pick up hydrogen ions and transport those hydrogen ions along with it and pick them up in the red blood cells and remove them from solution so they can't cause damage to other proteins in the red blood cells. So here we can see the hydrogen ion being picked up by the hemoglobin. Now the hemoglobin can do this because remember, I've taken the globin part, the protein part, this is the heme unit right here, the iron containing unit, and here's the globin portion, which is the protein portion. Remember, the protein is made up of a chain of amino acids. And periodically along this chain, there are amino acids that have side chains that are negatively charged side chains. So these negatively charged side chains that we see here represented by the little hooks will attract these positively charged hydrogen ions, and the hydrogen ions will link on to these negatively charged side chains and be pulled out of solution. Okay? Now, the hydrogen ions, we're pulling them out of solution because they can link on to other proteins and cause damage to those proteins. But what about the hemoglobin molecule? Well, the hydrogen ions, whenever they bind to the hemoglobin molecule, they do cause a change in the structure of the hemoglobin molecule. It causes a little shifting in the change of the hemoglobin molecule, and that affects the affinity of the hemoglobin for oxygen. This leads us to something that we call the Bohr effect. This happens in the systemic tissues out here we're talking about now. The Bohr effect says that the decrease in affinity of hemoglobin for oxygen is caused by the binding of hydrogen ions, which facilitates the release of oxygen into the tissues. So you may recall, whenever we were taking a look at the oxygen hemoglobin dissociation curve, that there were two additional factors other than the concentration of oxygen that affects the delivery of oxygen to the peripheral tissues. One of them was temperature, and we talked about how temperature affects that delivery. The other one was pH. Here we get a chance to see how pH affects oxygen delivery. Because as carbon dioxide enters these cells and carbonic acid is formed, it dissociates to form hydrogen ions and the pH begins to drop. These hydrogen ions now attach to the hemoglobin molecule, which is transporting oxygen. The hydrogen ions then cause a slight change in the structure of the hemoglobin molecule, and that structure of the hemoglobin molecule decreases the affinity of hemoglobin for oxygen even further. So in active tissues where there's a lot of carbon dioxide coming into the red blood cell, there's going to be a lot of hydrogen ions formed, and these hydrogen ions are going to be picked up by molecules of hemoglobin, and when they are, it's going to cause that hemoglobin to release oxygen even faster than it otherwise would. So the more active this tissue is, the more carbon dioxide it produces, the faster the red blood cells are going to release the oxygen into those tissues because of that pH change. Okay, well, we've talked about now 1, 2, 3 different methods of transport so far. We said there were 4 methods by which carbon dioxide is transported. So to review, a small amount of carbon dioxide is transported in the plasma. The remainder of the carbon dioxide enters the red blood cells, converted to bicarbonate ions. Some of those bicarbonate ions combine with potassium to form potassium bicarbonate. There's our transport method number 2. The remaining bicarbonate ions are transported out of the red blood cell, where they combine with sodium ions to form sodium bicarbonate. And the sodium bicarbonate is transported in the plasma. That's our third method of transport. Our fourth method of transport, then, any carbon dioxide that still remains that was not converted into carbonic acid will, in fact, also combine here at the tail end of this hemoglobin molecule. And it attaches to the tail end of the hemoglobin molecule to form carbamino hemoglobin. So that's our fourth method of transport. A small amount of carbon dioxide can be transported on the hemoglobin molecule as well. Okay, so these are the ways that carbon dioxide is transported via the blood. Okay, now moving ahead, we know how the hemoglobin gets into the blood and how it's transported, but now how is the I should say carbon dioxide, how is the carbon dioxide, whenever the blood gets to the lungs, how the carbon dioxide released? Well, before the carbon dioxide, I have CO, this is a real, it came out really, really tiny, but that's a 2. Before the carbon dioxide can diffuse out of the blood and into the alveoli, it first has to be released back out of that bicarbonate ion. For that to occur, bicarbonate ions have to combine with hydrogen ions to form carbonic acid again. Once that forms, the carbonic acid will quickly split then to form water and carbon dioxide. The carbon dioxide will then diffuse out of the blood and enter the alveoli. So essentially what's happening is the reverse of what we just looked at in the previous slide. So here's our previous slide here now again and everything that we've shown. So here's what's going to happen. Now let's say that this is a capillary bed, and this capillary bed is now associated with an alveolus. So now the blood is going to enter the capillary bed that surrounds the alveolus. Okay, and now what's going to happen is these cells now, let's say, are going to be the cells of the alveolus. And now what's going to happen is this carbon dioxide here, which was moving along its concentration gradient from the cells into the interstitial fluid, into the plasma, and then into the red blood cell, now here on the other side of the alveolus, the respiratory membrane, if you recall from our discussions in part 1, the carbon dioxide is going to be in a lower concentration across these cells than it is here in the blood plasma. So what's going to happen is, instead of the carbon dioxide moving into the red blood cell, it is now going to reverse and it's going to move out of the red blood cell. As that carbon dioxide is removed from the red blood cell, concentration levels go down, and now this carbon dioxide is released from the hemoglobin molecule, and that carbon dioxide continues to flow along its concentration gradient back out of the red blood cell, out of the plasma, out of the interstitial fluid, and across the alveolar wall, the respiratory membrane. When that happens, carbonic anhydrase not only can catalyze the chemical reaction which combines carbon dioxide with water to form the carbonic acid, but now this enzyme can reverse that process. Now the enzyme, under low carbon dioxide concentrations, will stop combining water with carbon dioxide to form carbonic acid, and now it will take the carbonic acid and split the carbonic acid to form water and carbon dioxide. So what happens is the carbon dioxide is going to continue then to move along its concentration gradient because it's being produced in the reverse process. So as that happens, as this carbonic acid starts to decrease, more carbonic acid is going to be formed now, because what's going to happen is the hydrogen ions are going to be released from the hemoglobin, and they're going to combine with, first of all, this bicarbonate ion from the potassium. So it's going to unhook from the potassium. It's going to combine with the hydrogen ions to form more carbonic acid. In addition, as the bicarbonate ion concentrations go down, this process is going to reverse itself, and the bicarbonate ions are going to start to move in the opposite direction along their concentration gradient. So the bicarbonate ions are going to pass through the band 3 protein and re-enter the red blood cell and continue to combine with hydrogen atoms to form carbonic acid, which will be acted upon by the carbonic anhydrase to be split into water and carbon dioxide, maintaining higher levels of carbon dioxide. So the carbon dioxide now will diffuse out of the red blood cell into the plasma, and from the plasma into the interstitial fluid and across the respiratory membrane into the alveoli. And when we exhale, we blow off that carbon dioxide. So everything's kind of happening in the reverse here. In addition, there are chloride ions here. Now that these bicarbonate ions are coming back in, the chloride ions will shift back to the outside to their original positions again. Okay, so now let's focus here for a minute. These hydrogen ions are coming off the side chains of the hemoglobin molecule. How does that affect the hemoglobin molecule? Well, once again, that is going to cause the hemoglobin molecule to shift back to its original shape. And when it shifts back to its original shape, this is going to result in something called the Haldane effect. The Haldane effect says that the binding of oxygen decreases the affinity of hemoglobin for hydrogen ions, thus facilitating the release of carbon dioxide from the red blood cells. So in other words, what happens here? These hydrogen ions are coming off the side chains. Why are they coming off the side chains? They're actually kind of getting pushed off the side chains, because now we're back up here in the lungs, and what's happening? Oxygen is being transported into this red blood cell, and oxygen is binding to the hemoglobin molecule. As the oxygen binds to the hemoglobin molecule, it's going to have an effect of pushing these hydrogen ions off the hemoglobin molecule. The hemoglobin molecule then is going to change its shape, and when it changes its shape, the binding of the oxygen is going to decrease the affinity of this hemoglobin molecule for these hydrogen ions. They're going to fall off. They're going to combine with bicarbonate ions to form the carbonic acid. So in the lungs, whenever we inhale, the actual flow of oxygen into the red blood cells, and as the oxygen starts to combine with the hemoglobin, it causes the hemoglobin to lose its ability to hold on to these hydrogen electrons, and the hydrogen electrons fall off, combine with the bicarbonate ions to form the carbonic acid, which then the carbonic anhydrase affects and causes it to split into water and carbon dioxide and keeps the flow of carbon dioxide going out of the cells. So this is how carbon dioxide is released from the red blood cells into the plasma and eventually across the respiratory membrane and into the lungs. Okay. All right, so what I'd like to do now is, and I don't know if you can see this all, but you'll be able to see it on your PowerPoint. I'd like to move on now and start to wrap up our discussions on the respiratory system. And in the first part of our wrap up, I'd like to talk a little bit about important lung volumes and capacities. So in order to speak about this, let's go back to our definition of respiration. Now remember, for an anatomist and a physiologist, we said that respiration actually has 3 distinct phases to it: ventilation, external respiration and internal respiration. But clinically, we said that respiration is defined as one inspiration and one expiration. So the physical act of breathing in and out. Okay, this is the clinical definition of respiration, not the anatomical definition. So when we talk about respiration, how many times on average does an adult, a healthy adult, actually breathe in and out? How many respirations occur per minute? Well, I have up here about 14 to 18 respirations per minute is the average for a healthy adult. Now you have to remember I come out of a sports medicine background. So you may have learned in your nursing classes or other clinicals that it's a wider range than that, maybe 12 to 20. That's because it takes into account geriatric patients. I'm taking into account mostly young, healthy athletes. So 14 to 18, 12 to 20 respirations per minute, the average number of respirations. The question becomes, as we breathe in and out, how much air is actually moving in and out of the lungs with each breath? We can measure the amount of air that's exchanged between the atmosphere and the lungs during these respirations with an instrument that's called a spirometer. So here on the right-hand side, you have one type of spirometer. This is called a dry spirometer. It has a dial at the top that you can see there that is calibrated, and it has a mouthpiece down here at the bottom. And that mouthpiece, the patient can put up their mouth, close off their nose, and breathe through that mouthpiece, and the dial will measure how much air actually passes through that spirometer with any one breath. Why is this important? Well, measuring lung capacities then can help us to diagnose certain respiratory diseases and disorders. So let's take a look at some various lung volumes and capacities that we can identify. The first one is tidal volume. The tidal volume is the amount of air that you breathe in and out of your lungs during normal, quiet breathing. The average normal tidal volume is somewhere around 500 milliliters. So, as you're sitting there right now, just taking notes, watching the lecture, you're just sitting there quietly and you're breathing in and out. How much air are you exchanging with each breath? That's called your tidal volume. You're sitting there, you're not exercising, you're not working hard, you're just going normal, quiet breathing. Well, the average normal tidal volume is around 500 milliliters. And that's going to change. That's across the board average. It will vary by gender, and it will vary by height, weight, and age. But generally, overall, it's about 500 milliliters. The next lung volume capacity we take a look at is what we call the inspiratory reserve volume, or IRV. That's defined as the total amount of air that can be inhaled over and above the tidal volume. So if you need to get more air in because you need to exchange more oxygen and carbon dioxide, you can increase the total volume of air that you can breathe in. That amount that you can increase by is called your inspiratory reserve volume. Next up, we have the expiratory reserve volume. That's defined as the amount of additional air that can be expelled from the end of a normal exhalation. So you're breathing normally and quietly, and now we want to know, beyond that tidal volume, how much more air could you blow out if you absolutely have to? That would be the expiratory reserve. Let me try to demonstrate it to you, and I'll exaggerate it just a little bit, but hopefully you'll get the idea. So if I'm doing tidal volume, I'm breathing in and out normal, quiet breaths. Now, if I want to look at inspiratory reserve, what I may do is I take my normal tidal breath in, and then I say, how much more air can I breathe in? So I would go, and that additional air above tidal would be my inspiratory reserve. On the other hand, if I take my normal exhalation of my tidal volume, and then blow out as much more air as I can, that's my expiratory reserve. So it may look something like this. And again, this is a little exaggerated, but the idea would be the end of my tidal breath, and then after the end of the tidal breath, how much more air could I blow out? That's my expiratory reserve. The next volume and capacity we take a look at is called the minute respiratory volume. The minute respiratory volume is defined as the total amount of air that enters and leaves the lungs during 1 minute of normal quiet breathing. So all we're really doing there is we're saying, okay, how much air are we taking in during normal tidal volume? Let's say it's 500 milliliters. How many respirations did we carry out in 1 minute? 10, 500 times 10, 5,000 milliliters, or about 5 liters of air moved in and out of our lungs during 1 minute of quiet breathing. That would be our minute respiratory volume. Okay, the next one we have is what we call vital capacity. The vital capacity is the maximum amount of air that can be forced out of your lungs after inhaling as deeply as possible. That's referred to as the vital capacity again, and the vital capacity of an average person is about 4,500 milliliters, or 4.5 liters. Okay, so the maximum amount of air that can be forced out of the lungs after inhaling as deeply as possible. Take as deep a breath as you possibly can, and then what we're going to do is, let's say, for example, with that dry spirometer, we're going to blow into the dry spirometer. We're going to see what's the maximum amount of air that we can blow out till we can't blow out any more air. That's our vital capacity. Okay, so here's a spirogram that shows how the different lung volumes and capacities work. Here on the left-hand side, we have the of the spirogram, and this is in liters, lung volume in liters over the course of time. So here we have the tidal volume. So this would be, I'd be taking my normal breath. Okay, and so that's going to be about 500 milliliters we can see between these two lines right here. Okay, now we have the inspiratory reserve volume. So I take my breath in, and then how much more air can I take in? That's my inspiratory reserve, from the top of the tidal volume to the maximal inspiration. Okay. On the opposite end of that, we have our expiratory volume. So now I breathe out, and how much more air can I blow out at the end of a normal quiet breath? There's my expiratory reserve volume. Okay. Now, we should mention here that down here at the bottom of the graph, there's this line right here, and this line represents residual volume. That is to say, the residual volume is the amount of air that remains in your lungs all the time. You don't really exchange it, so to say. It's just there, and it's keeping your lungs inflated. If you were to be able to blow out the residual volume, you'd collapse your lungs. So the amount of air that's in your lungs just to keep them inflated is called the residual volume. Okay. So over here, our next volume that we have here, we didn't kind of really talk about it. This is called inspiratory capacity. So the inspiratory capacity is the total amount of air that you have the capacity or the ability to inhale. So you would go from the bottom of your tidal breath, where you exhale, and then take in as much air as you can. Do you see the difference between inspiratory capacity and inspiratory reserve? Inspiratory capacity is the total amount of air you can inhale. Inspiratory reserve is just the amount of air that you can inhale above your tidal volume. Okay. Next up, we come across here, and we have our vital capacity. So what we do is we take a deep breath as we possibly can to get up to our inspiratory capacity, and then we blow all that air out that we possibly can. And hopefully we get down whoops, let me go back here a minute. Apologize for that. So we go back and we blow this out, and hopefully we can blow out all the way down to residual volume until we can't blow any more out or our lungs would collapse. That gives us our total vital capacity. Now, if we take the vital capacity and add to that the functional residual volume, we get the total capacity of our lungs, the total amount of air that our lungs can hold. And then finally, we have here the functional residual capacity. Okay, so this is the amount of air that actually most of the time stays in our lungs during normal quiet breathing. So unless we really try to blow out that extra air, this is the amount of air that's actually not being exchanged while we're sitting there in normal quiet breathing. So this is a typical spirogram that shows those different features. Okay. Okay. So we'll finish up with some respiratory diseases here. I've really kind of taken these diseases and written them out in pretty much detail, so there's not a whole lot more that I can say about them. But if you look on your exam review sheet, it is important that you be able to identify these respiratory diseases. So do take a look at them and get these notes down as you take a look at each one of them. So I'll leave each one of these up here for just a couple of minutes each so you can get the notes. So we have bronchitis, and the next one is pneumonia. And we can talk about bacterial pneumonia versus viral pneumonia and the differences between them. Then we have tuberculosis, a little bit about that. And pertussis, or whooping cough, which seems to be making a comeback these days because people are opting not to get the DPT vaccine, diphtheria, P, pertussis, and tetanus, T. Next one is pleurisy. This has to do with the serous membranes that surround the lungs we talked about in part 1. Asthma. Many of you may suffer from asthma. Cystic fibrosis. And again, most of these respiratory disorders are going to, in some way, affect those lung volumes and capacities that we talked about before. So whenever we take a look at those lung volumes and capacities, if we see any deviation from normal, it could lead us to a diagnosis of one of these different respiratory disorders. And emphysema. And I believe SIDS is our last one.